The glyoxalase system is a metabolic pathway important to sustain human health and an emerging target for improved treatment of disease. It catalyzes the removal of a potentially damaging metabolite called methylglyoxal, or Mg. It is comprised of two enzymes, glyoxalase 1 and glyoxalase 2, and a catalytic amount of reduced glutathione. Glyoxalase 1, or GLO1, catalyzes the removal of methylglyoxal and plays a key protective role. In aging and certain disease, diabetes, cardiovascular disease, obesity, there is abnormal accumulation of methylglyoxal in a state called dicarbonyl stress. Dicarbonyl stress has increased risk of methylglyoxal modification of protein and DNA, which is associated with cell dysfunction and mutation. The glyoxalase system is an emerging target for pharmacological intervention for improved treatment of disease. The glyoxalase system was studied by the very early pioneers of biochemistry over 100 years ago. Only in the last 30 years, however, with improved analytical techniques and methods in cell-based studies, preclinical and clinical studies, have we really understood the involvement of the glyoxalase system in sustaining health and disease. And the Thornalley-Rabbani team has been involved in nearly all of these developments and played a leading role in many. The underlying involvement of glyoxalase in sustaining health and development of disease is linked to the slow decline of glyoxalase 1 activity in aging and more rapid decline of glyoxalase 1, and sometimes linked to also increased production of methylglyoxal, particularly in metabolic and vascular disease. From the regulation of the glyoxalase system and its dysfunction in disease, we can begin to model the glyoxalase systems mathematically in a systems approach. The systems approach enables us to predict the response of the system when we intervene in it pharmacologically, so we can confidently develop drugs based on the glyoxalase system.
To define the status of glyoxalase system, the measurements required are the concentration of methylglyoxal, glyoxalase activity and expression. Expression can be measured either by mRNA or protein, but preferably both. Other measurements required are Mg flux formation by measuring D-lactate, Mg-derived adduct content of protein and DNA, glutathione and S-lactol glutathione, GLO2 activity, mRNA and protein, also required to define the status of glyoxalase system. Rabbani-Thornalley team have developed, validated and optimized these techniques in their lab, and the latest technologies they have developed are the imaging of Mg using fluorogenic probes and screening technologies to screen GLO1 inducers. Standardized protocol for all these techniques are available from their lab. By assessing the glyoxalase system, we can identify patients who are suffering from dicarbonyl stress and enable us to develop a personalized care. The timeline to develop GLO1 inducer functional food is 1 to 2 years. Timeline to develop GLO1 inducer nutritional medicine is 3 to 5 years. And the timeline to develop GLO1 inhibitor for anti-tumor, anti-malarial and antibacterial drug is 5 to 10 years.
The glyoxalase system is ripe for expansion and exploitation to make new functional foods, pharmaceuticals, and improved personalized health and care. We are looking for partners and investors to help develop these technologies and bring them to market.